All right. So before we get started with what I did last summer presentations, first, thank you all for being here. I do really appreciate it. On top of that, I want to let you know about the next event that is going to be happening as a part of the Scott Science Scholars STEM event series, and that is Tom Gilmore. He is an engineering manager at Evanik in Etowah, Tennessee, and that's a company that has locations in like 13 or 15 states around here and abroad as well. So a lot of opportunities there if you're interested in engineering. He's going to be talking about the different opportunities at his plant specifically and some of those more broad positions that may be available. That's October 24th, same time, same location. Okay, the last thing I want to tell you before we get started with this is hold all of your questions for the end. That way everybody has the same amount of time to speak. And then if you need to write it down, just write it down. We'll get back to it at the end. All right. So I'm going to introduce our first speaker. This is Mr. Chance Loveday. He is a sophomore computer science and mathematics major. He was invited to be a part of the SULI program last year. That is the Science Undergraduate Laboratory Internship, and that it takes place at Oak Ridge National Laboratory. Today, he's going to be talking about the design and implementation of a radiation transport code input generation interface.
Thank you, Brett. As he just said, I was an intern in the SULI program down at Oak Ridge National Lab over the summer. And my project was designing and implementing a radiation transport code input generation interface. And I did my research under my two mentors, Dr. Douglas E. Peplow and Dr. Rob Lethervier. So I interned in the radiation transport group under the Nuclear Energy and Fuel Cycle Division down at Oak Ridge. And my project focused on making an application that would take information from various databases that had material composition information, such as its like atomic mass, its isotopes, its different compositions, and would generate input that researchers could use when using different modeling softwares. And these modeling softwares, they would use instead of actual physical experiments because of the physical risk. So instead of experimenting at nuclear facilities and running these tests manually, they would run it through these modeling systems that would model the behavior of radioactive materials as it would react inside of a nuclear reactor. And some applications of this, for instance, would be radiation shielding, which is important, right? If you live in a community that is near a nuclear facility, you don't want the radiation leaking out and causing health effects, any unwanted health risks. So my project can be traced back to 2012, where it was picked up by another intern who created a graphical user interface in Java. And he made it compatible with two different databases for two different systems. And it worked great as a standalone in Java. But my mentor wanted to restart the project so we could make a more intuitive interface that was compatible with the more modern versions of the modeling systems that they're using today. And I'll talk a little bit more about WASP in a minute, but it's a collection of readers and parsers and different components that I needed throughout this project. So this project read in different pieces of data from databases. And for those of you who are familiar with databases, I didn't use MySQL or anything of that nature. My databases were strictly in JSON format. And just as you can see up here, it's this type of format where you have a token assigned to a different value. So for instance, I would have a database that would be assigned to an array of different materials, which would thus have different components. And I would pull information using the tools from WASP. And my job was to write a program that would be able to utilize these resources, that would use these readers and parsers and actually read in and assign objects using C++. And the reason why we swapped over to C++ instead of Java is, for one, because it is a compiled language, so it compiles everything at once and it's much faster. So there were three pieces of software that were crucial in this project. First of all was GitHub. It's a software that they use in industry for collaborative use. So I would upload my code to a repository and my mentors could measure my progress that way. And I could see the different versions of my code as I added new updates to it. So if I were to mess up somehow, there were different tests that were running on what's called pipeline. And if something causes my code to break, or if something causes a test to fail, I can go back and trace back that issue rather than having to do that manually. And furthermore, I use Google testing, and it's not just typing into Google and testing it that way. It was a header package that I included in C++ that was inside of WASP that I used from there, where I would run different various tests. So for instance, like loading into databases, I had to actually load them and assign values based on what it read in from the JSON. And I had it structured in a series of true or false statements. So if something didn't meet a requirement, if a material didn't have a name, or if a material didn't have components, then it would fail and it would tell me and my code would break. And the WASP tool set included a different set of readers and parsers and other tools like Google testing that I used throughout my project. So I ran into a lot of syntax errors and logic issues as I began this project, and that was to be expected because I knew basic C++ going in, but I only had about 3 to 4 weeks of prior experience in the language, and I had to transfer a lot of Java logic to C++, and it's not a clean conversion. So I'm going to break down different pieces of my project. So my main focus was in a file that I called material.db, which is where I handled reading in from material databases. And there were 4 main parts that I had in this program. I had to figure out, one, how am I going to define these objects as I read them in? So I noticed in the JSONs, I could split them into 3 main objects: a database that had a series of materials, materials that had properties like name, what type it was, and it had a list of components, which is the 3rd class I used, that told me what element I was using and what its count was, or atom count was, or what its fractional composition was. And I also used an optional contact class that were in a couple different databases that I used. Secondly, I created a series of what I call build methods, where I would physically go in and it would assign those values from the JSON it pulled using the reader and parser, and it would assign those values and fill in those objects. So that's what I would use in my testing to load the database so I could work with this information. And then I would have methods that would go and check to make sure the data I was using was actually correct. So for instance, I was working with a lot of fractional compositions. So I would want to make sure that those fractions added up to 1 with a very low % error. And I did catch 2 or 3 instances where I had to go back and clean up some of the data due to round off error. And that was a good means of checking that. And finally, the end goal we're trying to get to is formatting the input for the users, that they can actually go in and use this input in their modeling systems. And when I say input, I'm talking about I want to specify for a certain material at a certain type and at a certain amount. So if I wanted to use, let's say, for example, uranium nitrate, I want to say I want uranium nitrate, and I want them in weight fractions, and I want 100 grams. And then you would tell the program that I want to use this, and you could see how it behaves over time. And next, I had to access a masses database because I was working with various isotopes, especially when you're working with radioactive materials such as uranium and plutonium, you're working mostly with their isotopes. So I created a masses class that was very similar to the material DB, but it was structured more so as a masses database that had elements with different isotopes. And then I had a program that I called Fate GUI that was meant to simulate a user interface where the user would interact with the program, and you could give it various commands. You could say, I want to change the material. I want to get an input code. I want to look at the information that is listed with this material, and you could do several different options. And I put the parts in place for a GUI, but my mentor and I did not have time to actually go and construct a GUI. So this is what we used for testing purposes. And then finally, the actual testing portion of my project, I used Google testing and statements such as assert true and assert near to test what my program is actually outputting and what the correct value should be. And I use these tests to generate correct values that I can use for further testing. So as my results, I came out with about 2,000 plus total lines of code, and I helped contribute to the creation of a convenience tool that will hopefully be widely used across the national labs. And the project is now waiting to be picked up by the Nuclear Code Integration Group, and will hopefully be integrated straight into the modeling systems interface rather than being a standalone. So I wanted to show you what a project like this would look like. This is an open source software called Ultimator Cura. So this is Cura's GitHub repository. So you have all the different files and folders. And then, as you can see, the little sidebar, there's a lot in there just based on the scroll bar. And here's what a file would look like in C++ using C++ code syntactically. And here's my poster. And I would like to thank both of my mentors and Oak Ridge National Lab SULI program, as well as the Department of Energy's Office of Science, for allowing me to conduct this research over the summer. And I'd also like to give a special thanks to Dr. Jeff Nichols and Dr. Shiopsis for recommending me this internship. And before I wrap up, I'd like to spend a couple minutes talking about my personal experience. 30 seconds. 30 seconds? Okay. So going into this internship, I was a freshman, didn't have a whole lot of computer science coursework. So to say the least, I was overwhelmed. And a lot of people I talked to didn't recognize Maryville College. They'd heard of UT, but they hadn't heard of Maryville College. But I found this opportunity a good source of professional growth, and I was able to grow my confidence in my personal ability, and I made a lot of new connections. And finally, I wanted to spend just a few seconds talking about a optional summer craft.
And as you, another quick round of applause. Thank you. So as you can imagine, there's a lot of security risk potentially with code related to Oak Ridge National Laboratory. Therefore, we're not seeing any actual script from Mr. Loveday today. And next, as I pull this up, so I want to introduce this next speaker, Mr. Alonso Angel. And I want to just say, I think this is a fantastic representation of what a liberal arts education can do for you. Mr. Angel is a senior outdoor study and tourism major. But this past summer, he applied and got accepted for a Jim Hill Fellowship at the Tennessee Aquarium and also worked with the Tennessee Aquarium Conservation Institute directly to do a lot of just a lot of very interesting things. His talk is going to focus on what he did during his time there. And without taking up any more, turn it over to him.
All right, so hi everyone, I'm Alonso, and I'm talking about my summer at the Tennessee Aquarium. So I worked at the Tennessee Aquarium Conservation Institute. So this is the conservation department of the Tennessee Aquarium, and I'm sure many of you have probably seen the aquarium, been there. So I actually worked in a separate building. I didn't work in the actual aquarium where you see all the cool exhibits where kids go. So located in Chattanooga, Tennessee, and they pretty much work to protect and conserve fish all over the Southeast U.S. So as you mentioned, I was a Jim Hill Fellow. That was my position. I worked from May 15th to July 28th. And so big thanks to Dr. Crane for actually sending me this position last spring. I don't think he's here, but big shout out to him. And so my job was kind of support all their ongoing projects and studies. I didn't have necessarily one study or one project the whole summer. So it was kind of cool because I got to do a little bit of everything, learn a lot of cool skills, and just learn a whole lot of cool stuff. And so I worked with fish all over Tennessee, in Alabama, Kentucky, and in Georgia. So now I'm going to talk about a few of the studies and things I did this summer to aid them in their work. So the first one was a thermal tolerance study. So this one was done in collaboration with a PhD student from Canada. Her name was Brittany. She's a really cool person. And so we tested the thermal tolerance of sand darters all over the Southeast. So sand darters are small little fish species. You can see them right there on the screen. There's a lot of them in the bucket. And so they're called sand darters because they like to bury in the sand. So we used these big green seine nets. We kind of dragged them through the water to catch these sand darters. And once we caught them, we kind of had this setup right here and run through these trials. And so this little box right here where the sand darters went, we put them in there. Once they're in, we started that big mechanism up there that kind of heaved the water up slowly more and more and more. And so once these sand darters flipped over, we would pull them out of the water and we record the temperature, and then we would measure their length and their weight and put them in their recovery bucket where they would kind of assimilate back to the water that they're used to, eventually be returned back to the river, back to their home unharmed. And so this is really cool to the travel for this project. We spent 4 days in Kentucky in the Red River and the Green River, and then 4 days in Alabama in the Coosa River. So the next one, the Sequatchie River Project. This is a very big project the aquarium is doing in collaboration with other big organizations. This is actually a 5-year project that was started this summer. And so the Sequatchie River, located here in Tennessee, is a pretty impaired river. It's got high levels of E. coli, a lot of agricultural runoff. And so this big 5-year plan is kind of meant to help improve the river, try to clean up the water, and implement better agricultural practices to help improve the river. And so our role in this project was kind of get a baseline of what fish are found in this river. And so we, once again, used a big seine. We went through different parts of the river, sang through it, sampled, and try to find what fish were there. So we recorded like how many species of each fish were there. And so right here, you can see us working, sailing through the river. And then these two fish are kind of two of the cooler fish we found. The top one is a blenny darter, and the bottom one is a water paint shiner. And these are pretty common throughout Tennessee. You can find these actually in the little river right outside campus. Next, propagation. So at Tennessee Aquarium, they actually raise 3 fish species. They raise tangerine darters, brook trout, and lake sturgeon. And so the brook trout, probably my favorite. They are actually our only native trout here to East Tennessee. And so we raised those. And the cool thing about those is I actually got to release the brook trout this summer as well because they did grow to the appropriate length to be released into the wild. And so you can see in the middle two pictures, that's us releasing the brook trout back into the wild. They were released into mountain streams in the Cherokee National Forest. So about an hour and a half south from here. On the right are the lake sturgeon. So when they arrived, we got about, I believe it was over 1,000 lake sturgeon. They're tiny, tiny, tiny. And when they first arrived, we would spend hours and hours chopping their food because they had tiny mouths and were really delicate. And so those are actually going to be released this month into the Tennessee River. And then the bottom left are the tangerine darters. Those are also actually found here in the Little River, right outside campus. These are really cool, clever fish. And these actually have a kind of unique project. They're actually not being released to the wild, but instead, once they get to a certain length, they're being shipped over to another facility in Middle Tennessee where they're being used to raise freshwater mussels. This one's probably my favorite we did so far in summer, was the bridle darter snorkel survey. So the bridle darter is a small fish. It is endangered to Georgia. And so we did a 2-day snorkel survey in North Georgia. Day one consisted of snorkeling the Consuelo River. So this is all these pictures are from our day one. We got these canoes and kind of went down the Consuelo River, and we stopped in certain areas of the river where we thought was ideal habitat for bridle darters. And then day two, we did not a canoe, but we just kind of snorkeled upstream in an area called Talking Rock Creek. That's right. And so we kind of snorkeled upstream. And then after 2 days, we only found one bridle darter, which kind of shows why they're in danger in Georgia. But overall, I thought it was super fun because we're just snorkeling, having fun, and we saw so many cool fish. So lastly, the MARC recapture study. This was the biggest study I did, probably, or I didn't do it. I was involved in the one I was most involved in. And so this one started in April, so a month before I started, and it is still ongoing to this day. And so in Tennessee is a little area kind of south of Crossville, if you know where that is. It's called Walden Ridge. And inside Walden Ridge is a creek named Duskin Creek. And so there's a culvert on Duskin Creek. You can see it on the bottom right corner. That's the culvert. As you can see, it is pretty beat up and damaged. So the aquarium is planning to replace that culvert in next summer of 2024. And so the plan is that they want to conduct a study to figure out how replacing this culvert will affect fish movement through the stream. They want to know whether it would improve it or would make it worse. And so for our study, this was the site for our study. There was 3 pools in the creek. There was pool A, B, and C. So pool A, I guess kind of see the colors now, but pool A is the pool downstream. Pool B is found right below the culvert, and pool C is upstream of the culvert. And so pools A and C are reference pools to be compared to pool B. So for this study, we had to tag fish. We used VIE tags, which stands for visible implant elastomer. So what we did is we kind of use these needles you see on top right, and we inject them into the fish, kind of tag 2 of these fish. And we tag a total of 450 fish, 150 per pool. So we tag during the months of April, May, and June. And then after their tag there is released. And then the tag is very important because it's unique to the pool and the month the fish was tagged in. So you can see on the left side, the red tag, for example, is red on the right side, would be like April in pool A. So this is very important when recapturing these fish so you know where these fish are tagged from and how far they've traveled up or downstream. So now recapturing. So to recapture these fish after they are tagged, we use the big green seine and we pull the net through inside of each pool. And we also seined 100 meters upstream and downstream of each pool. And so this was done monthly. And this was probably the most fun. We just kind of pulling through, catching a bunch of fish. And obviously, most of the fish stayed in the pool that they were originally tagged in because fish just don't move as much as other animals do. But we did find some fish that did move up. I got to make a lot of cool connections in the community. I mean, thanks to this opportunity, I'm actually presenting at a conference this November on that MARC recapture study. So overall, it was a really cool job. Thank you.
All right, so I am introducing our final speaker today. This is Ms. Hannah Phillips. She is a junior biology major, and she completed her internship this summer, or her summer research project, using funding from the Fund for the Improvement of Post-Secondary Education grant that was received by the Scott Science Scholars Program from the Office of Congressman Tim Burchett. And we're going to find all about what Hannah did, detecting and isolating Naegleria fowleri in Northeastern Tennessee waters.
All right, so as Brett said, this is the project that I got to work on over the summer. So I got to help on this project with Mackenzie Nicholas and Dr. Bergotti as our advisor. And so my part that I helped with over the summer, we really did a lot of environmental sample collecting and optimizing of our protocols, but it is an ongoing project that Mackenzie is still working on. So lots of things beyond what I did, but just a little bit of background information. So Naegleria fowleri is also known as brain-eating amoeba, which is pretty scary sounding. But it is primarily found in very warm freshwater, such as rivers, ponds, lakes, in the rare case, also tap water. So the reason why looking for this amoeba is so important is because it obviously causes a very serious infection. It can cause a PAM infection, which basically occurs when someone is swimming in contaminated water or they like jump into it and the water goes up their nose. And then that could reach your brain and that could destroy your brain tissue. So obviously very serious, but it is also very rare. There's only been about 200 cases worldwide since 1965, but most of them were fatal, having a mortality rate of 97%. So a little bit more about Naegleria. They have a life cycle which consists of cysts, trophozoites, and then a flagellated form. And the form that we were most concerned with was the trophozoite form because that is their feeding and most active stage. So they can only infect you when they're in this stage. If they're in the other two stages, then they're not going to infect you because that's the stage they go to conserve resources when nutrients are scarce. We also had looked at previous research that showed that the trophozoites would create feeding trails when grown with Alonso E. coli. So that's really what we relied on to look at through microscopy. So we could possibly see were there amoebas making trails in our E. coli. And if there weren't, then obviously we'd move on to the next sample. So what we did at each of our sample sites is we collected 100 milliliters of water from 5 different locations around the area. And then we pulled that all together for a total of 500 milliliters that we were working with. If the water had a lot of debris, then we tried to filter that through cheesecloth to get that out of there. And we also recorded the water's temperature and pH at every site. Here's a table of all the sites we went to over the summer. As you can see, some of them are very local, even in our own college woods and the green belt. But then some of them we went a little bit further away to the Knoxville and Kingsport area. And then here are just a few pictures I took while we were sampling of some of our sites. You probably recognize some of these. That bottom middle one is at the green belt. And that top far corner is at Meads Quarry, which is in Knoxville, if any of you guys have been there. So once we took our water samples, we quickly tried to get them back to the lab for processing. And that happened through a vacuum filtration. So as you can see up there, that's kind of how we set up the filtration system. Originally, that circle piece of filter paper was completely white, and that's how it looked after it went through the water at the Alcoa Duck Pond. So very green. But then once we got that filter paper, we shook it in a tube with sterile water to try to get the contaminants off that paper. And then we centrifuged the tube and we resuspended the pellet that was formed in saline. And then we plated that suspension onto our non-nutrient agar plates, which had a lot of E. coli on them. And that E. coli was there for hopefully the amoeba to eat and for us to be able to see that. And then those were incubated overnight. So, and a lot of aspects of this research had to do with microscopy. We did a lot of microscopy because every sample site had 2 different plates that had to be looked at daily. So plates accumulated very fast. And what we were looking for were those feeding trails. So we kind of did a control plate with our culture of Naegleria fowleri to kind of see what we were looking for. We ended up having so much amoeba on it that, as you can see of that first picture, that's just a whole line of like the amoeba just like eating the whole plate of E. coli. So we were having a bit of trouble with the microscopy of what we were truly looking for. There wasn't a whole lot of published research on what these feeding trails look like, especially with the microscopes that we had in the lab. But here are 2 of our environmental samples under the microscope. As you can see, especially this middle one, we've got a lot of crazy curved lines going. But then some of our samples had more like singular, just like thick lines. And so trying to decipher what was a bit of a struggle. But once we were kind of confident that we thought some of these were feeding trails, it was time to extract the DNA from them. And so we kind of had a couple of different techniques that we tried with this. The first being scraping the cells off of the plate with an inoculating loop. And then we shipped those into a centrifuge tube and began the process of following our DNA extraction kit. That didn't yield as much DNA as we were hoping for. So then we tried another technique called flooding the plate. So that's where we actually opened up our plate and we poured sterile water in there. And then we wrapped it with parafilm so we didn't spill anything out, and we shook the plate to hopefully get all those cells into the water. And then we use that water to then centrifuge and go through our DNA extraction protocol. And then after that, we would do a PCR to amplify the DNA. And then we would run those PCR products on a gel to see if we got a band for the correct size of DNA we were looking for. But that is about where I stopped in the process of the summer of my part of the project. And Mackenzie is still testing all of our environmental samples. So now I just kind of want to share some outcomes and some lessons that I learned. I think this was a really valuable experience. I obviously learned a lot about safety because we were working with something that was deadly, but it was going to be pretty hard for us to infect ourselves injecting it up our nose, but we were still very careful. We worked under a biosafety level 2 lab, and we took all those precautions. Sterile technique and things like that was a great learning experience. Also just troubleshooting. Sometimes things didn't work and we didn't know why. And so we had to kind of problem solve and figure out what worked, what technique didn't. And that was very beneficial, not just for research, but just in general, learning how to deal with problems and challenges when they arise and when things don't go the way that is planned. So yeah, overall it was a great, very beneficial experience. Thank you for listening.
All right, we can go ahead and bring our other speakers back up and we will open the floor for questions. We have about 10 minutes here. Alex, you want to start us off?
You said level 2 safety lab. How many levels are there? Which is the most dangerous?
I believe there's 4 levels, 4 being the most dangerous. We did not know. We had to work under like a biosafety hood whenever we had the Naegleria out, and we had to have gloves, lab coat, and goggles. But that hood has like a glass layer to like, and only your hands go underneath, so it can't like splash in your face or anything like that.
Okay.
So in order to have a supercomputer, were you able to work with a supercomputer during your internship?
So my project did not involve the supercomputer. I did get to take a tour and see the supercomputers, and I got to work as part of HPC. I got to work on a node cluster called Ascent at Oak Ridge, but I didn't get to work with any of the really fast supercomputers like Summit or Frontier.
How long did you work on your project, Chance?
So I was down in Oak Ridge for about 10 weeks. And as part of SULI, we have other requirements where we have to like send in a report, send in a poster, and we had other seminars we had to attend. So I really only worked on my project about 6 to 7 weeks out of those 10 weeks.
For Alonso, when you were injecting the fish with the elastomers, how long do those last? Like, how long are they going to keep sampling these fish? How long does that take to break down?
So those will last about 2 years. So yeah, they last a good bit. So they'll last the entire study.
Yeah.
Chance, did you know C++ before you started this?
I had self-learned, I knew about what I could learn in about 3 to 4 weeks prior to. I had self-learned C++, and I was a little self-sufficient in it about a year before going into the internship, and I used about 3 weeks before to brush up on it.
And question for you. So you mentioned there's not a lot of research about this topic. Why did you guys start to investigate?
I didn't really choose that. That was more Mackenzie and Dr. Bergotti. But I think that they saw where it is such a rare thing when you get infected. However, with the global temperatures kind of rising, I think that we're seeing more cases than we have before because waters are warmer than normal. So I think it was just kind of the prevalence of the alarmingness of the increasing rate. Yeah.